Hello everyone, welcome to Dokuz Eylul University Meet Sidra Medicine webinar series. We have two distinguished speakers today. Before introducing them, let me ask you to unmute your microphones, and also if you have any comments or questions, please write in the chat box. We'll have those at the end of two talks. The first speaker is Dr. Patrick Tang, who is the Division Chief of Microbiology at Sidra Medicine and Associate Professor of Pathology and Laboratory Medicine at Weill Cornell Medicine. He got his MD PhD degree in British Columbia, Canada, and medical microbiology specialty training at University of Toronto. He has fellowship of the Royal College of Physicians of Canada, in addition to his clinical role at Sidra as a consultant medical microbiologist. Dr. Tang is responsible for the development and implementation of new molecular and genomic-based tests for infectious disease at Sidra Medicine. Prior to joining Sidra Medicine, Dr. Tang was the clinical scientist leader at the British Columbia Centre for Disease Control and clinical associate professor at the University of British Columbia. He has taken on many roles from the bedside to the laboratory bench to epidemiology in past global outbreaks, including SARS outbreak, also H1N1 influenza pandemic, as well as we know COVID-19 pandemics. Dr. Tang's clinical and research interests are in the application of genomics and metagenomics to infectious disease and public health, as well as the development of novel molecular diagnostics. Patrick, floor is yours.
Thank you very much for the introduction and the invitation to speak today. So today I'm going to be talking about using wastewater as a method for surveillance for viral infections. So I have no financial interest to disclose. And today we're going to talk about the utility of doing wastewater surveillance for viruses, some of the uses of it during the pandemic, as well as during FIFA World Cup, which was just over here in Qatar a few months ago, and the use of extending this concept to doing surveillance for avian influenza. So we know that surveillance is a very important part of the public health management of infectious diseases. Surveillance is necessary to understand how much of that disease is present out there and the epidemiology of it, so that we can adjust all of our interventions in public health, as well as inform vaccination strategies. We need to monitor the trends so that we know if our interventions are working. And of course, we need to identify outbreaks and any novel pathogens that might be emerging. So we've known this for many hundreds of years. People have known that the environment is an important part of infectious disease surveillance. Back in the United Kingdom, they knew in the 19th century that the River Thames was the source of the cholera and typhoid. So there's two main ways of doing surveillance. One is event-based or case-based surveillance, and that's where you look for people that have been infected or hospitalized or have died from an infectious disease. And you might modify some of the case definitions based on, what symptoms might be encountered or what lab tests are positive. But the dependencies for this type of surveillance is that the population of your country must have access to healthcare and access to laboratory testing. And you also have to have a very good system for gathering all of this data, monitoring, and looking for trends that are going to trigger alerts for you to take action. The other way of doing surveillance is go into the environment. So this is sort of independent of the things that we do in terms of our health-seeking behavior. One way to do that is to test wastewater. And the advantage of something like wastewater is that you can detect asymptomatic as well as mildly ill people who wouldn't otherwise go to see a doctor or go to the hospital. However, it is complicated because you have to understand the pathophysiology of the disease. When does the virus peak in your different samples? How much is shed in your stool, in your urine? And then after it gets into the wastewater, what are all the different factors that influence the persistence of that organism in the wastewater? So there have been lots of examples in the past few decades where we've used wastewater to do surveillance for pathogenic viruses. So the obvious ones are ones that are fecal orally transmitted, like all the gastroenteritis and hepatitis type viruses like Hep A, Hep E, adeno-astro-norovirus. But people have now been extending it to other viruses. So we know respiratory viruses, many of them can be detected in wastewater, as well as other viruses, things like Ebola, enterovirus, HIV, measles can all be detected in wastewater too. So the problem with using wastewater surveillance is that it may not always correlate with some of the case-based epidemiological data. And there's many reasons for this. So the wastewater collection, the boundaries of the sewage and how it's collected might not match, your healthcare administration or political type boundaries. We might not have very good case data for a lot of diseases because we don't do a lot of reporting for many infectious diseases. So we don't really know the true incidence or prevalence of many diseases. And of course, there's many physiological and environmental factors in the environment that could affect the detection of these specific pathogens. So the basic steps in doing wastewater surveillance are, we have to go collect a sample. You could either grab one single sample or do a composite sample over a period of time. You could try to get the water part of the waste or go for the sludge. And then the next step is that you have to concentrate. And there's many ways to concentrate, from filtration, centrifugation, or flocculation, or precipitation. And then you have to have a good way of extracting the viral nucleic acid out without bringing along all the inhibitory substances that are going to block your assay. And then you need to measure the amount of viral nucleic acid, whether you use PCR or doing a next generation sequencing method. So we know from the COVID-19 pandemic, testing was one of the very important cornerstones of managing the pandemic. And very early on, we had to start collecting all the case-based data. So you remember when there were only thousands or tens of thousands of cases back in early 2020. And now, towards the end of the pandemic, we're close to almost 700 million reported cases and almost 7 million deaths. So if we look more closely at all of this case-based surveillance, there are many gaps in the data that probably don't accurately reflect the true incidence of infection. So if you look at Turkey, there's a huge gap after you guys stop reporting your cases after a certain time. If you look at Qatar, there's weird spikes or drops in the data when due to weekends or holidays or things like that. If you look at United States or Germany, UK, definitely underestimation of the huge Omicron outbreak that happened at the beginning of 2022. And then China, of course, the data there is not very accurate due to many different reasons too. So we know that case-based surveillance could highly underestimated the true number of COVID-19 infections out there. There's many reasons to that. Some people might have not be able to get testing, especially at certain times where there's different policies that restricted people from getting tested, or you have to pay for the test. So that could be a barrier to testing. There could be many biases in the data because certain areas have better access to testing, or certain people might have go for testing because they need for travel or for work. There could be incentives that make people want to get tested because they don't want to go to work. There could be penalties against some people, so they want to avoid testing. And then at one point, we started doing antigen testing at home. So all these people started testing themselves at home instead of going out to a hospital or clinic to get tested. So then all of that data is potentially lost. And many regions didn't have data sharing agreements, so it was very difficult to collate the data in certain countries. And of course, when the peak of testing, we didn't have enough capacity or resources. There were huge delays, from days to even weeks delay in testing, so data wasn't accurate. So one alternative to this was looking at wastewater. And in Qatar, some of my colleagues here had developed a very robust wastewater surveillance program. They went to all the different wastewater treatment plants and collected the wastewater on a weekly basis. And you can see from this graph here on the right that there was a pretty good correlation between the blue, which is the case-based surveillance data, against the red, which was the wastewater viral load. And you can see that the spike in the wastewater actually preceded the peak of the clinical cases in the hospitals and clinics here. And if you extend this more to other viruses, so there's an example here from Santa Clara County in California, where they started testing, well, since we have these samples, why not look for influenza, the other coronaviruses, RSV, parainfluenza virus. And they found that there was also good correlation of these other viruses with the positive percent positivity of the other viruses based on case-based detection. And this was also useful for informing not just their COVID-19, but also other respiratory virus public health decisions. And if you extend it even more, here's an example from the University of North Carolina in Charlotte, North Carolina, where they did this at the university campus level, where they collected wastewater from the different dorms. And they were able to detect small clusters of cases in their university that weren't detected by case-based surveillance and self-reporting. And they even found a single asymptomatic person that was shedding virus in one of the dorms. So there's a lot of power in using wastewater surveillance if you apply it to different levels of the wastewater collection system. So during COVID-19, we found that wastewater surveillance could be used as an early warning system, because the levels that you saw in wastewater often preceded the hospital data. It helped public health officials direct the resources to where it was needed. You're able to test entire communities in an unbiased manner, as well as communities that might be underserved. And it was a really good way to check your case-based surveillance data and make sure that it was actually accurate as well too. And you can also use it to track the different variants in your communities. So the advantages was that this type of testing was non-invasive, so no one had to be poked in the nose. It was inexpensive, and it's also scalable. It's unbiased, and it tests both symptomatic as well as asymptomatic or mildly ill people who wouldn't otherwise seek medical attention. And it was useful for showing community level changes in the infection rates and allow us to correlate this with other measures of the COVID-19 pandemic. So the next example I wanted to show is about polio. So we all know that there's a global initiative to get rid of polio. We've already eradicated 2 of the wild type polios, type 2 and type 3, and now there's only wild type type 1 polio virus that continues to circulate in Pakistan and Afghanistan. So we know that the oral vaccine is the most effective vaccine against polio, but it contains a live attenuated virus. And sometimes that live attenuated virus can develop mutations that allow it to spread in the community and then cause clinical cases of polio. And we also have the inactivated poliovirus vaccine, which is not quite as effective, but it does protect people against serious disease, but does not block the transmission of poliovirus. So in terms of poliovirus management, there's 2 avenues of surveillance. One is looking for acute cases, so doing surveillance for acute flaccid paralysis. The problem with doing this is that it's very inefficient. So 99% of these people that are kids that have acute flaccid paralysis are negative for poliovirus. And we also know that when you look at people that are infected with polio, only less than 0.5% of people infected with polio will develop acute flaccid paralysis. So this is a very inefficient way of doing poliovirus surveillance. And from us that work in the lab, we know that it's very hard to collect stool samples. We're always chasing people for stool samples. Easier to get blood, easier even to get an LP and collect a CSF sample, because collecting stool is difficult, it's unpleasant to handle, and the timing is difficult because you never know when the bowel movement is going to come. So this is especially of concern because there's been recent poliovirus detected in the West here. So in New York, in London, and Jerusalem in 2022, there were cases of poliovirus infection, as well as detecting outbreaks in the community. So there was one case in Jerusalem, one case in New York with paralytic poliomyelitis. And we know, of course, this only reflects a small percentage, a fraction of a percentage of the real cases of infection in the community. So wastewater surveillance was initiated after these cases were detected in those regions. And then they found that there was widespread circulation of vaccine-derived poliovirus in those communities. So that allowed public health officials to then initiate poliovirus campaigns in those regions to stop those outbreaks. And one tool that hasn't been used as much as it should be is a novel oral poliovirus vaccine, which has emergency use authorization, and that contains a more stable form of the poliovirus type 2 that is less likely to cause outbreaks. So this was especially of concern to Qatar because we had the 2022 FIFA World Cup towards the end of the year in 2022, and the WHO was very worried that this mass gathering would then lead to transmission of poliovirus in our country. So we took up samples that were part of the COVID-19 wastewater surveillance program. So the 5 wastewater treatment plants were now sampled on a more frequent basis, so twice weekly instead of weekly. And these were processed, and the samples are sent to my lab to then test for poliovirus. So fortunately, during the FIFA World Cup, before and after the FIFA World Cup, we did not detect any poliovirus in any of the samples of wastewater from all the different sites in Qatar. However, one of the other things we detected, we were looking for many other things at the same time, like foodborne viruses, bacteria, as well as enteroviruses. So we did notice a big spike in enteroviruses right in one of the communities right before the start of the World Cup. And that was really most likely due to a huge influx of all the workers and all the visitors that came in to help with the setup of the World Cup activities. And we also noticed this also correlated with a spike in enterovirus infections detected in young adults, which were most of the people that came in during this time. So in terms of poliovirus surveillance, wastewater surveillance is a well-established tool for the global eradication efforts. Most of the countries that are polio-free don't really do this on a regular basis, whereas places like Pakistan have very advanced and comprehensive systems for wastewater surveillance for poliovirus. When you do detect it in the wastewater, it indicates that there's a high level of person-to-person transmission of poliovirus in the community. And it's especially important if there's communities within a country that have low vaccine coverage. Or in communities where there's a lot of connections, travel connections to regions that are still endemic with polio or frequently have polio outbreaks. And when we detect this, it's a good way to allow public health officials to then target their polio vaccination campaign efforts. And the final example I'm going to give is avian influenza. So we know this is in the news right now as one of the things that we're very concerned about. Influenza is really a bird virus. It probably emerged about in 8000 BC in birds, And then over time, it's eventually spread to humans. Probably the first cases are recorded in human history around 400 BC. And then later with the advent of farming towards the end of the 19th century and early 20th century, it then spread to poultry and swine. So the evolution of avian influenza viruses is facilitated by the fact that birds mix very well. Just like humans have a very good network for global travel, birds also have this. So if you look at all the migratory bird flyways around the world, there's many places where birds can mix and mingle together, and that helps with the evolution of avian influenza viruses. So we know that right now there's a huge outbreak, probably the worst outbreak of H5N1 ever in birds. So this is a highly pathogenic avian influenza virus clade that emerged around late 2021. It's caused huge mortality in many bird species. So when birds are infected with, wild birds are infected with this, the mortality rates are around 50–60%. It's spilled over into probably over 100 different mammalian species, from bears to foxes to skunks to dogs and cats, and even marine mammals like dolphins and seals. There have been sporadic outbreaks in poultry. So at least 140 million chickens and ducks have been killed by the virus or culled because they've been infected since October 2021. We haven't had outbreaks in the swine population yet. And so far, we've only had 11 reported human cases, which is very fortunate that the virus hasn't picked up mutations that allow for efficient human-to-human transmission. So we know that the normal way of surveillance is very resource intensive. So capturing wild birds and then trying to swab them, right? And very stressful for the animals. So I'm going to talk about an outbreak that I was involved in 2014, 2015 in Canada. So in many parts of the Western world, we have high density poultry farms. So if there's any incursion of a virus like an avian influenza virus, it can be very deadly to these the birds that are in these farms. So there were 13 chicken and turkey farms that are affected by H5N2 and H5N1. These are only avian type viruses, not pathogenic to humans. So during these different incursion events, during that time period, they had to cull a lot of the birds and institute a lot of quarantines. So our idea was to do sediment sampling. So same as collecting stool and the sewage from humans, why not collect the stool and from the droppings from the birds? So we went to where the wild birds congregate to all the water sites where we see lots of wild birds, and we collected the sediment samples from there. And then these are the samples we get in the lab. So there's dirt, there's twigs, there's all kinds of stuff. And we were trying to get this into a PCR machine or a next generation sequencer. So it's very difficult, dirty work. So we found that there were a lot of inhibitors. So as you would imagine from that sample there's tons of inhibitors. So a huge rise in your CT values for PCR. And our strategy was then to do next generation sequencing. So we wanted to characterize the hemagglutinin and neuraminidase segments from the viral genome to see if we can detect the outbreak strains in the sediment sample. So we had two strategies. We weren't sure which was going to work. One was just doing a whole bunch of PCR amplicons across the viral genome. The second was to do a capture hybridization method to pull out the sequences and then do amplification and sequencing. So the first strategy is basically blanket the genome with PCR primers and hopefully pull stuff out and then sequence them. Second strategy was then to try to pull out all the influenza sample DNA away from the inhibitors and all the other types of non-influenza DNA and RNA and sequence that. So if we compare that now to conventional surveillance, which is, looking for dead birds, so either hunters have killed them or dead birds that are found on the highway and other places like that. So prior to the outbreak, none of the wild birds tested positive for H5. During the outbreak, there were only three positive birds that were detected, but none of the sequences from these wild birds that were incidentally found contained the same H5 sequences that were affecting the farms that had the outbreaks. Later, after the outbreak in 2015, there was definitely more surveillance in wild birds, and just about 1% of them had that H5 sequence detected. So if we look at doing PCR on all those sediment samples, the positivity rate from those wetland samples are about 7.7% for true PCR positives. So really count those low positives, which probably had PCR inhibition. We have another maybe 16% that were positive. We then target the outbreak farms where we looked at all the waste and the ponds and other things in the outbreak farms at a high rate of positivity. So 51% positive, another 30% low positive. So definitely lots of influenza RNA in the waste from these affected farms. So if we do sequencing, the PCR amplicon strategy didn't work at all. None of our methods could overcome all the inhibitors in the samples, but the target capture method was very successful, but didn't correlate very well with PCR. So we got out of 48 samples that we sequenced, 26 had been PCR positive. We got 19 of those. 9 were PCR low positive. We were able to take 5 of those 9. And from 13 that were PCR negative, we were able to find influenza sequences in 8 of them. So in those 19 samples that were positive for H5, we could see that 13 had outbreak strain 1 and 3 had outbreak strain 2. And then others had unrelated H5 sequences that were in the wild birds, and other samples had other subtypes of avian influenza viruses are not related to outbreaks. And just is just a heat map showing all the different H and N sequences that we recovered. So if we then focus on the outbreak farms, so all the ponds and other things that are from in the farms, we had 11 outbreak samples. 3 were PCR positive, 8 were low positive by PCR, but all were positive for avian influenza virus reads. And 7 had outbreak strain 1, 1 of them had outbreak strain 2, and then 2 of them, we weren't able to detect the outbreak strains. We detected other unrelated avian influenza viruses. And also at other points near the farms also had a high rate of positivity for one of the outbreak strains. So if we then look at all the sequences, all of the sequences from the sediment samples and the outbreak samples from the affected chickens were highly related. There's only 2 SNPs that separated them, so they were all part of the same subclade of influenza viruses. So we were able to show that doing sediment surveillance was much superior to the conventional strategies for surveillance. The wetland sediment sample surveillance was very successful. PCR wasn't so good because of all the inhibitors, but we used targeted resequencing where we captured the sequences, pulled them out. We had a very good recovery rate of the outbreak strains in our sediment samples. And comparing that to conventional surveillance where you look for dead birds, that was only able to detect, we only found the sequences in 1% of the dead birds. So very highly inefficient, and none of the outbreak strains were actually detected. So what's next for wastewater surveillance? So this is a very important strategy, as we found from the COVID-19 pandemic. So we need to leverage some of these established systems that we've done during the pandemic to other communicable diseases. And the other problem with many of the things that we've done during the pandemic is many of them are voluntary, many of them are not funded. So we also have to find funding from the government to then assist all of these programs to continue testing for COVID-19 and other infectious diseases, and also extend it to other places that might be more important as well too, like airports, zoos, wildlife parks, and mass gatherings, such as international sporting events, where we might need to have similar surveillance data. So with that, that's the end of my talk. I thank you for your attention, and I can take any questions if there's time for that.
Thank you, Patrick, for your excellent question. I wrote at the chat box that we are going to have the questions and comments at the end of the both talks. So let's move to the next speaker. Professor Arzu Sayıner is currently faculty member in the Department of Microbiology at Dokuz Eylul Üniversitesi School of Medicine. She is directing molecular microbiology laboratory at Dokuz Eylul University Hospital. She received her MD degree from Ege University. She finished then specialty training in clinical microbiology and infectious disease at Ege University as well. She worked as a visitor scientist at Mayo Clinic, United States, and Erasmus Medical Center, the Netherlands. Her research areas include essentially medical virology in terms of diagnostic nucleic acid assays. She is the member of a number of societies like European Society for Clinical Virology, European Society of Clinical Microbiology and Infectious Disease, also Turkish Society of Microbiology and Turkish Society of Clinical Microbiology and Infectious Disease, and so on. She has advised 9 theses so far, published many articles, over 150s, and got citations over 600. She has contributed to books and book chapters and proceedings, and many projects either published or ongoing. Professor Sayıner will talk about applied metagenomics in diagnostics, microbiologist's perspective. Dr. Arzu Sayıner, floor is yours.
Thank you very much for this nice introduction. I also would like to thank Dr. Tang for the excellent presentation regarding wastewater surveillance for viruses, which was possible with the help of next generation sequencing. So he showed us the importance of this new method and how it can be used for virus surveillance by using different samples from the nature. And I will continue to talk about next generation sequencing, and I will talk about the clinical use of metagenomic sequencing, especially in the diagnostic microbiology. So I have no financial disclosures. Now, when we say clinical metagenomics, maybe we can describe it as using NGS on clinical samples to improve diagnostics and management of infectious diseases. And the ultimate goal is, of course, to help clinical decision-making and improve the patient outcomes. My goal with this presentation is to answer these 3 questions: when to apply metagenomic sequencing to clinical practice, what are the most beneficial clinical uses, and what are the advantages and limitations of metagenomic sequencing. In the current state, infectious disease diagnosis relies on either direct or indirect detection methods. And traditionally, diagnostic microbiology most depends on...
Erken ölüm riskini konuşalım. Erken ölüm riski yaratan durumlar nedir? Tabii ki bu noktada
I think somebody's microphone is...
I unmuted her.
Okay, thank you. Thank you very much. So I was just saying that although traditionally microbiology, diagnostic microbiology depends on culture, It can take many days and sometimes weeks, depending on the pathogen. And also there are several factors that can influence the sensitivity of the culture. Although it is a very important, very powerful diagnostic tool for detecting viable microorganisms, still the sensitivity depends on many factors. For example, preemptive antimicrobial therapy can decrease the sensitivity. If there are unculturable or fastidious pathogens present in the sample, those are difficult to, sometimes impossible to culture. Sampling and storage problems also cause sensitivity decrease for culture-dependent methods. And therefore, up to 50% of the infections are culture negative today. So we need other methods in order to improve the diagnostics in infectious diseases. Now we have a very powerful tool that is metagenomic NGS. It can provide huge data, and that can be used to detect, to identify microorganisms, to detect their genotypes, to detect the virulence markers. We can analyze microbiome, which is present in the sample, and we can detect new pathogens, and therefore we can use the method for outbreak investigation. And also this huge data provides information regarding antimicrobial resistance genes, and it can be used to characterize host-immune responses, and therefore can be used for differentiate infections from non-infection. Sometimes it's very important not only to diagnose infection, but separate it from non-infectious situations. So the metagenomic testing starts with choosing the patient's sample. It can be blood, CSF, tissue sample, many types of samples, a variety of samples can be used for metagenomic testing. And the next step is extracting the nucleic acid. This includes DNA and RNA if you want to cover everything. And if extracted RNA is also going to be used for the sequencing, then it has to be converted to complementary DNA, of course. And this mixture, this extract, includes a mixture of host and microbial nucleic acids. And therefore, it will be important to aware of this, this mixture for the bioinformatic analysis. The next step is the amplification and library preparation. In this step, adapters and barcodes are added to the nucleic acid fragments in order to allow sequencing and multiplexing. And finally, the last step is the sequencing, which will provide a huge sequence data. And it is necessary to use bioinformatic tools to analyze this data. Otherwise, it's not possible to get a result out of this huge sequencing data. Now, when we look at the bioinformatic analysis, first a quality filtering has to be done in order to get rid of the low quality reads. And then the host genome subtraction has to be done. Human reads can be used for further analysis, such as transcriptome profiling, genome analysis. So it is not totally useless, but it will not be used for detecting microorganisms. So at the end, after quality filtering and after host subtraction, we have the data belongs to the microorganisms, which has to be analyzed against the chosen microbial databases. And then finally, by doing this, we can classify, we can describe the pathogens which are present in the sample, including the read numbers and the coverage rates. The final result report is prepared and provided to the lab, to the clinician. So those are the main steps of workflow for metagenomic sequencing. Sensitivity of the metagenomic sequencing depends on many factors. In fact, all the steps that I counted can be modified in order to increase or decrease sensitivity or specificity depending on the desired result. But the most important factors affecting the sensitivity of metagenomic sequencing is the amount of microbial nucleic acids present in the sample. We all know that typically the amount of microbial nucleic acids are higher in the early stages of infection, and therefore the sampling time is important for providing a better result. And often the amount is related to the infection severity. We also remember that symptoms can persist after microorganisms is cleared from the sample, and therefore some samples could be very challenging to detect a pathogen. The other factor affecting the sensitivity is the amount of host DNA or RNA which is present in the sample. If this amount is large, then it can decrease the sensitivity of metagenomic sequencing for detecting microorganisms. And therefore, acellular fluids such as plasma could be a better choice for choosing the sample type if we want to increase the sensitivity. Of course, it all depends on the infection. So the metagenomic testing is briefly, it's target independent, it's culture independent, and it is not affected by prior antibiotic use. And it can be directly applied to the clinical specimen. In fact, metagenomic sequencing can identify more potential pathogens in a patient sample than any single conventional assay. So we expect a big clinical impact from such a powerful diagnostic tool. It can reduce morbidity, mortality, hospital stay, ICU admissions by providing a better diagnosis. It can contribute to more effective and more narrow spectrum antibiotic therapy, and therefore help to reduce the resistance problem. And it can also avoid additional and sometimes invasive procedures, which might be used for the diagnosis, and therefore reduce the cost of healthcare and help us to better control the infections in the hospital, in the healthcare settings, and in the community. So the expectations are several. However, it is still a costly diagnostic tool. It requires a special infrastructure, expertise. It lacks standardization. Therefore, it is difficult to use it as the first-line test. We have to choose the target. Maybe we should start by asking where conventional methods fail. The reasonable target populations could be immunocompromised patients, since the number of microorganisms, the type of microorganisms that can cause infection in such patients could include a wide range. And also patients with central nervous system infections, with sepsis, prosthetic joint infections, or with fever of unknown origin can also be among the target populations, since it's difficult to diagnose the infections in such patient groups. And also patients using empiric antibiotics are one of the target groups, since the culture methods based on culture mostly give false negatives. So as I mentioned, patients with central nervous system infection is one of the target groups, since the etiology is unknown up to 50% of the cases. And there is a large variety of potential pathogens, especially for immunocompromised patients. The sample volume is limited. Therefore, it will be very desirable to detect the pathogen with a single assay. Today, widely used assays are the syndromic nucleic acid tests. Those are the multiplexed PCR assays. They are fast, but the spectrum they cover is quite narrow compared to the metagenomic sequencing. They can only detect up to 15, 20 pathogens. Now, in this study, this is a multicentral prospective study where they looked at 204 patients. CSF samples were tested by conventional methods and metagenomic sequencing. And when the results were compared to the clinical decision, sensitivity was 80%, specificity was even higher, 98%. detection was only possible by metagenomic sequencing in 13 of the 58 infections. And in 8 of those, the result, the metagenomic sequencing result, changed the therapy. So it had a clinical impact. However, there were still some false negatives, even by metagenomic sequencing. And those patients had to be diagnosed either by serology or by using samples other than CSF. And the main problem was the low titers of pathogens. So the pathogen reads were below the pre-established reporting thresholds. And that was the reason for false negativity. So this study showed that metagenomic sequencing improved diagnosis of CNS infections and provided actionable information in some cases. However, the highest diagnostic yield resulted from the combination of metagenomic sequencing and conventional testing. Another study, this is a single-center retrospective study, if the patients were critically ill or culture negative, their CSF samples were tested by MNGS, and those were all community-acquired infection, patients with community-acquired CNS infection. And the results show that 42% of the patients were infected with non-culturable pathogens. Some of them were viruses. And altogether, the metagenomic sequencing detected 41 microorganisms, and 32 of them were not included in the film array panel, which is the widely used commercial syndromic multiplex assay today, widely used in the diagnostic laboratories. When the reads of the pathogens were compared, the lowest reads were detected from the patients infected with Mycobacterium tuberculosis. So Mycobacterium tuberculosis infections provided the lowest number of reads, and this sometimes could be a problem for making a diagnosis. So difficult to diagnose pathogens were detected with reads between 14 to 290,000, and among those, there were some unique or unexpected pathogens such as Streptococcus suis pseudorabies virus. This study showed that metagenomic sequencing identified a wide variety of pathogens, some of which could not be detected by conventional methods. In fact, this is the result we expect from metagenomic sequencing. Another study from France, in this study, 52 CSF samples were analyzed by culture, film array panel, and Oxford nanopore sequencing. With some adaptations, this sequencing tool, this metagenomic sequencing assay, provided a very fast pathogen identification. With only 20 minutes of sequencing time, it was possible to identify the pathogen in 33 samples. In fact, in all the positive samples, the result was available in 2 hours of sequencing time. The total workflow took about 4 hours or less than 4 hours. So the study showed that metagenomic sequencing even could be used in the point of care setting. It also provided genotyping and in silico antibiogram for bacteria, for cultured and uncultured bacteria. And 62% of those results were in concordance with phenotyping assay. So that's another important additional information. Now, in this picture, we see the metagenomic sequencing corner of the point of care laboratory. They were able to set up the laboratory in only a few square meters, and they provided results in a rapid way. Another problematic infection for central nervous system infections is the Mycobacterium tuberculosis meningitis. It's difficult to diagnose. The culture takes several weeks. Therefore, WHO recommended a commercial nucleic acid assay as the initial test for tuberculosis meningitis because rapid diagnosis and rapid initiation of treatment is essential. In this single center retrospective study, they analyzed the CSF samples by 4 different methods for tuberculosis meningitis. They used midget culture, modified Zille-Nelson staining, expert assay, this commercial assay recommended by WHO, and finally, metagenomic sequencing. When we look at the specificity and the positive predictive value, all of those 4 methods provided a perfect result. It was 100%. But when we look at the sensitivity, the best result was provided by metagenomic sequencing, and it was 63%. When we compare it with expert nucleic acid test, it was only 55%. So there was about 10% difference between the 2 methods, and the lowest sensitivity provided by the midget culture as expected. So this study showed that metagenomic sequencing outperformed expert assay, staining, and culture in CSF-based diagnosis of tuberculosis meningitis. However, it cannot be used as a rule-out test since the negative predictive value was only approximately 70%. So MNGS combined with expert assay or staining, modified staining, in order to enhance the sensitivity. Another group of infections where we have difficulty of diagnosis is the prostatic joint infections. Although mostly the pathogen is bacterial, mostly gram-positive bacteria, still the culture is negative up to 15% of the cases. And there is a very difficult treatment for such infections. Either a long-term antibiotic has to be used or some repeated surgeries has to be performed to treat such infections. So it's a problematic area, and the result given by the laboratory makes a big difference. In this meta-analysis, they looked at 10 studies, more than 565 patients. 1/3 of those patients were culture negatives. Metagenomic sequencing provided a sensitivity around 93%, specificity around 95%, and overall diagnostic value was almost perfect. So metagenomic sequencing detected pathogens in 54% of the culture negative patients and 90% of the culture positive ones. So there were a few false negatives still. Metagenomic sequencing is an important tool for detecting new pathogens, especially viruses. There is no common gene for virus screening. Unlike bacteria, we don't have that tool. Therefore, detecting unexpected or novel pathogens is important, especially for detecting new epidemics or pandemics. Therefore, the best example for this purpose is the detection of SARS-CoV-2 from patients with pneumonia in China in 2019, but that's not the only example. Similar to SARS-CoV-2, MNGS was used for detecting chikungunya virus outbreak in Bangladesh, Zika infections in Brazil, and Ebola virus outbreak in Congo. So those are just a few examples showing that clinical metagenomics not only improve individual patient outcome, but also provide earlier warning signs to improve pandemic preparedness. So that's another important advantage of metagenomic sequencing. Such unique or unexpected pathogens can be found, can cause infections in solid organ transplant recipients or in general immunosuppressed patients. This is a case report from France. It's a 61-year-old female patient who had a heart and lung transplantation 17 years ago. She was receiving maintenance therapy for immunosuppression, and she had a history of several infections caused by different pathogens. And finally, she was diagnosed by cytolytic hepatitis. Routine diagnostic methods all gave negative results. And finally, a liver biopsy was evaluated by metagenomic sequencing. And the result showed that the pathogen was a circovirus of unknown species. This was also confirmed by qPCR and in situ hybridization after the result received from the metagenomic sequencing. So as seen in this example, metagenomic sequencing can be used to detect unknown or novel pathogens. Another area for metagenomic sequencing is detecting pathogens in cell-free samples. There is a validated assay for plasma samples. It's a commercial send-out test which can detect more than 1,200 microorganisms. It gives the results with quantitative units, which helps the interpretation. So it is a non-invasive assay and a very attractive option to diagnose bloodstream infection. It calls the Carius assay. And this study, they looked at the clinical impact of this assay in 80 patients. Nearly half of them were immunosuppressed. The reason for ordering Carius test was mostly to detect the unknown pathogen. But in the other reasons, the other purposes was to avoid invasive procedures, to confirm the result, and to provide the early diagnosis. And the assay provided diagnosis in 65% of the cases where a positive impact was detected in 43. It was most beneficial for immunosuppressed patients, patients with sepsis, or patients who have received antibiotics less than 7 days prior to assay. The primary positive impact was the de-escalation of antimicrobial use. Another impact of this assay was studied in 140 bacteremia patients. In fact, in those patients, both blood culture and the Carius test repeated in every 2 to 3 days. Sensitivity of the sequencing was 90% almost, and specificity was a little bit lower, 74%. But the important finding of this study that the duration of metagenomic sequencing positivity was longer than the blood culture positivity duration. It was 15 days to 2 days. And when the duration was became longer, it was associated with the presence of metastatic infection. So the study showed that metagenomic sequencing was able to detect culture-negative endovascular infections, and patients with persistent positivity were mostly had metastatic infection, and they required additional evaluation. So, another area metagenomic sequencing can provide information is the antimicrobial resistance. So the resistance genes can be detected by metagenomic sequencing. It can both identify and let us to understand the antibiotic resistance mechanisms using either sequence or function-based approaches. And therefore, it can lead to more precise use of antibiotics and help us to overcome a very important problem, and that's the resistance. There are several databases specifically created for this purpose. However, the complexity of the relationship between phenotyping and genotyping antibiotic resistance test results limits its use, and accuracy varies by pathogen, antimicrobial agent, and metagenomic sequencing methods. So standardization is still lacking. We definitely would like to have a common conclusion, but that's not easy because there are variabilities between the MNGS studies. We mostly concentrate on the analytical phase of the studies, which is an easier part of the assay that can be controlled, but pre-analytical phase and post-analytical phase are as important. Testing indication, patient selection, choosing the sample type, and the timing of testing are all important factors which can change the result of the assay. And then when we look at the post-analytical phase, interpretation of the result, selected reference standard for the diagnosis, and criteria used for assessing the clinical impact are all also important for reaching a result. So all these factors can influence the result of the method, and therefore makes it difficult to have a common conclusion. Standardization and quality assurance are essential to implement a new method for routine diagnostic laboratory. This is also true for metagenomic sequencing. We must know that metagenomic sequencing can have problems such as false positivity and false negativity. The real cause of the infection must be distinguished from contaminants or microorganisms of no clinical significance, which can be detected by metagenomic sequencing. So interpretation is very important. On the other hand, if the absence of infection, if this knowledge is provided by metagenomic sequencing, it is also very, very important because it can prevent unnecessary invasive procedures or antibiotic use. However, metagenomic sequencing sometimes less sensitive than targeted methods. Therefore, false negativity is a possible problem. Therefore, interpretation of the result is very important. Negative results may not mean patient is infection-free, and the reported organism by the metagenomic sequencing method may not be the cause of the infection. Therefore, each result has to be evaluated according to the clinical context and the local epidemiology. And it will be great if a multidisciplinary team can help the interpretation of the result, which includes clinical microbiologists, infectious disease specialists, and the primary physician. There are several groups trying to produce create recommendations in order to implement metagenomic sequencing to the diagnostic workflows. A network of next generation sequencing of the European Society of Clinical Virology in one of those groups. And they already published two recommendations, one for the wet lab procedures, the other one is for bioinformatic analysis and reporting. And those recommendations or such guidelines, very important in order to make a better use of this very powerful tool. It will be great if we can have a validated testing provided by an accredited laboratory. This can definitely support the widespread use of metagenomics, enable better comparison of the results. It is available in some parts of the world, in some countries such as United States, and definitely having such an assay available will help the laboratories and the clinicians. In summary, clinical metagenomics have some advantages and disadvantages. It is target independent, culture independent, not affected by prior antibiotic use, and can detect pathogens, including unknown ones, and analyze functional genes. So it's a very powerful tool, but still requires expertise. It's costly, lack validation and standardization, at least in the current state. And the criteria for diagnosing the pathogens are not universal. Therefore, it requires a very careful result interpretation. In summary, I would like to emphasize that the accurate and timely results are very important for diagnosing infections. Metagenomic sequencing can improve individual patient outcomes and better prepare us for epidemics and pandemics for providing the diagnosis. There is a continuous improvement in the field, and standardization will definitely increase its clinical impact. In the meanwhile, diagnostic stewardship is important for optimizing testing algorithms, when to use metagenomic sequencing, when to add to this assay to the available diagnostic methods is important. And guidelines for clinical use are needed for rational applications. So thank you very much for your attention. If there are any questions or any contributions.
Thank you very much, Arzu, for your excellent talk. Looking at the chat box, there are some thanks I saw so far. Any question? No. Please, I would ask participants to ask a question. We can have maybe one or two questions. While waiting, can I ask a question to Arzu?
Sure.
Okay. Me and Patrick is working at Sidra, which is a pediatric hospital, tertiary pediatric hospital. So could you comment on specifically pediatric infection disease in terms of application of metagenomic NGS?
Well, maybe once again, central nervous system infections could be the first group of patients where we can have a maximum impact from the metagenomic sequencing. In our practice, 50 or even 60% of the samples sent to us, CSF samples sent to us for detecting a pathogen, for diagnostic work, are negatives. We cannot find any pathogen in those samples. We don't know if they are really pathogen free, or if the assay that we are using, or the diagnostic methods we are using, are unable to detect the pathogens. And in fact, there are a variety of pathogens, and currently we don't have any diagnostic tool for arboviruses, for example. We know that they are present in our region, but we don't have any available assay in the diagnostic laboratory to provide the result in a few days. Therefore, maybe metagenomic sequencing could be advantageous for detecting pathogen for central nervous system infections. I don't know, maybe Dr. Tang can make a contribution to this question.
Yeah, that's an excellent question. So, like my lab and my teams have been doing this for more than a decade, right, of metagenomic sequencing. And at Sidra, we've been doing this for the past 5 years routinely, metagenomic sequencing. So our experience is that in children, at least the population of children that live in Qatar, don't do all of those things that allow them to catch rare and unusual diseases. So definitely in adult samples, we see a lot more of the unusual things, but definitely in children, they tend to have the common things. And even the immunocompromised children, the things that we see in them are still common things that, because they don't go and try to experience weird things around the world and catch weird diseases, right? So in children, it's a different population, but definitely for young adults and other people that may be exposed to more rare or uncommon or novel pathogens, then that has a greater utility. So I think that was an excellent review of metagenomics. Pretty much everything that has been done in the past, all the advances in the past decade, and I will reiterate all the challenges. So the challenges are a lot, pre-analytical, getting the right samples, but post-analytical is extremely challenging. The bioinformatics is not easy, both not just false negatives, but false positive samples. So we play a large role as microbiologists in curating and interpreting the results from metagenomics so that we don't tell someone that they've got Ebola, right? So because you might have a inadvertent sequence that's detected, and that's actually happened in our experience. So yeah, so it's gone to one time we detected Ebola in a sample. So we went all the way to talking to all the manufacturers of the reagents, the controls. Some of our samples, we spiked in controls. And in one of the manufacturers that made one of the controls, in their factory, they also made controls for Ebola for other purposes. And we wondered if they might have contaminated our controls with a tiny bit of Ebola control, but they said it was not possible. So it was left at that. We found one Ebola sequence in a sample, and we even called up the patient, called up the doctor just to make sure there was nothing else wrong with it. But that ended up probably being a false positive or probably contamination from one of the reagents. So it can lead to a lot of misinterpretation, but so there has to be a lot of clinical judgment in using clinical metagenomics, at least at this stage of the technology.
Yeah, May I ask Dr. Tang, how long does it take for you to provide the result with metagenomic sequencing? May I ask when the results are available for the clinicians?
So for us, unfortunately, it takes a couple of days because we use the Illumina system and we prefer to do full sequencing of it. And we decided, okay, so there's different strategies. Like you said, there's the strategy of doing a lot of processing up front to try to remove the human and other sequences from the sample before you go on to NGS. And that takes a lot of work too, versus just trying to sequence it as deep as possible and hope that you find some pathogen sequences in there. So that's our strategy, less work, but maybe it takes a little bit longer. So then we can't stop the sequencing run early. We have to go all the way. So it does take us a couple of days. And we haven't really used the nanopore system. I know other labs have used that a lot more, but that hasn't been something that we've focused on. Our strategy has been more focused on using the Illumina system, which we're more comfortable with and more comfortable in terms of the bioinformatics. So definitely, as you said, there are examples using the nanopore, Oxford Nanopore, where they can generate results a little bit faster. And if there's a higher concentration of the pathogen present, they might be able to stop it early when they have enough reads that indicate the pathogens. But in many cases, the concentration is very low. So you probably have to go to the end, run the whole thing before you can analyze it.
Yeah. And also one last question. When do you go to the metagenomic sequencing? Who make the decision, clinicians or the microbiologists?
So the problem is the cost. So it's a combination of the clinician and the microbiologist that approves it. So, when you order one of these tests, whether it's UCSF, which are many of my friends there that you showed their pictures of, or Carius, whatever, it costs about $2,000 US to do it, right? So that's a big part of the test. It's very expensive still to do it. So you have to be very selective in terms of which samples you do. So there has to be a lot of approvals in place. And so it's still not that practical because of the cost and the time and the resources and the bioinformatics that's required to interpret the samples. And you have to do it very quick because infectious diseases, happen much faster than, other types of diseases. So you've got to get an answer fast and give an answer that's clinically relevant and not a false positive or false negative. Right?
Yeah. Thank you very much for sharing your experience. Thank you.
I also thank both of you for your excellent talks. Also, I would like to thank Professor Erdoğan and Professor Olgun for organizing these seminars. And I would like to thank all the participants joining to the seminar webinar. So we are ending the session right now. Hope to see you in the next seminar.
Thank you.
Thank you.
Bye-bye.